Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to all. We will begin our event with a prayer for grace led by Dr. Shahidi Zainal Abidin from the Faculty of Science and Marine Environment, UMT.
Al-Fatiha. Bismillahirrahmanirrahim. Alhamdulillahi Rabbil Alamin. Wassalatu wassalamu ala asyrafil anbiya wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma innaka khalaqus azim, innaka sami'un alim, innaka ghafurur rahim, innaka rabbil arsyil azim, innaka barul jawadul karim. All praise to you, Ya Allah, the Almighty, most great and most merciful, to you who create the earth and the heaven and all that lies within. All praise to Allah for what He bestowed us from the bounty of Islam and taught us wisdom and guidance from Quran. Ya Allah, may we ask you to give us good and to protect us from the evil. Ya Allah, we gather here today for the third seminar on biological security and sustainability 2021. With your grace and blessing, this program shall run as planned. Through this humble supplication, we express our gratitude for this opportunity and we seek from you, Ya Allah, endless guidance and blessing in our present and future endeavors. Ya Allah, the most merciful and most forgiving, give us the strength to perform the task ahead and to fulfill the responsibilities that are associated with them. Inspire us to adopt positive attitudes and protect us from adopting habits that deter progress. Ya Allah, you encompass everything in mercy and knowledge. Therefore, forgive us for our wrongdoings and grant us good in the life and hereafter. Rabbana alaika tawakkalna wa ilaika anabna wa ilaikal masir. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar wa qina adzabannar wa qina adzabannar. Wa sallallahu ala sayyidina Muhammad wa ala alihi washabihi wasallam walhamdulillahi rabbil alamin.
Thank you, Dr. Shahidi, for doa recital. Professor Datuk Dr. Asma Ismail, President Academy of Sciences Malaysia. Professor Datuk Dr. Aieni Haji Mokhtar, Vice-Chancellor University Malaysia Terengganu. Associate Professor Dr. Marina Muhammad Arifin, Dean Faculty of Science and Marine Environment, UMT. Associate Professor Dr. Mariam Taib, Chairperson of BIOSES 2021. UMT principals and senior officers, respected keynotes from Tianjin University, China, University of San Agustin, the Philippines, from Aligarh Muslim University, India, and from University Malaysia Terengganu. Our invited speakers from Kongunadu Arts and Science College, India, from Universitas Indonesia, from Évry University, France, from Heritage Environment Sendirian Berhad, from Malaysia Genome Institute, and from University Malaysia Terengganu. Seminar participants and guests, members of the Organizing Committee of BIOSES 2021, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh, and a very good morning to all. Welcome to the opening ceremony of the third National Seminar on Security and Sustainability Biology, BIOSES 2021, with the themes Facing Challenges Towards Advancements in Biosecurity and Sustainability. Organised by Research Interest Group RIG, Faculty of Science and Marine Environment, UMT. This seminar is conducted as fully virtual due to COVID-19 pandemic from 25th until 26th of January 2021. And thank you all for this great initiative in trying times such as now. Our most gratitude to Allah the Almighty for His grace and permission that we are able to gather in the ceremony of the event. BIOSES 2021 aims to provide a platform to look at the current research being carried out, thus promoting and encouraging among researchers related to security and sustainability biology. On behalf of the organizing committee, I would like to welcome all to today's event. It is a great honor to have you here. Next, we are showing you UMT corporate video prepared by the organizer. Gentlemen, I now call upon Professor Datuk Dr. Aieni Haji Mokhtar, University Malaysia Terengganu Vice-Chancellor, to say a few words of welcome.
Rahmanirrahim. Alhamdulillahirabbil alamin wassalatu wassalamu ala asyrafil anbiya wal mursalin wa ala alihi wa sahbihi ajmain. Thank you very much, our MC for the day, Dr. Maimah. To our special guest of honor, the Honorable Professor Datuk Dr. Asma Ismail, the President of Academy of Science Malaysia. Welcome, Prof. Datuk. Associate Professor Dr. Marina Muhammad Arifin, the Dean of Faculty of Science, Marine Environment, FSSM of UMT. Associate Professor Dr. Mariam Taib, the chairperson of BIOSES 2021, our respected keynote and invited speakers, as mentioned by our MC just now, from the University of St. Augustine, Philippines, Tianjin University, China, Aligarh Muslim University of India, Ganggunadu Arts and Science College of India, Universitas Indonesia, every University of France, Heritage Environments Sdn Bhd, Malaysia Genome Institute of Malaysia, and University Malaysia Terengganu. To our principal management team, conference organizers, honorable participants, guests, our lecturers and students, our sponsors, and the media group. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to all of you. Ladies and gentlemen, it is indeed my pleasure, my greatest pleasure, to extend a warm welcome to all the delegates, speakers and participants to the third seminar on Biological Security and Sustainability BIOSES 2021. Our praises to Allah subhanahu wa ta'ala, the Almighty, for we are given the opportunity to gather this virtually in the era of new normal for this auspicious event. We are very fortunate and grateful to have with us today my dear friend, Professor Datuk Dr. Asma Ismail, the President of Academy of Science Malaysia, the former VC of USM as well as USIM, to officiate the third BIOSES today. Being a specialist scientist, inventor, and a biologist herself, her research expertise in the health sector, as well as her vast experience as an inspiring thought academic leader, contributing to the higher education of Malaysia, as she was the former Director General, and herself being excellent in research, commercialization, and management in general. I would believe surely this gives a boost and the right person to officiate this seminar. BIOSES is one of the major activities hosted by BIOSES Research Institute Group, BIOSES RIG, which is recently established in the Faculty of Science and Marine Environment of UMT. The RIG started off with the international conference, initially with the Cell Signaling Conference, ICSC, in 2017, followed by first BIOSES seminar in 2018, and the second BIOSES seminar in 2019. Unfortunately, in the year 2020, we had to bring forward the third BIOSES seminar to today. We have gathered hundreds of participants from all over the country and also abroad. For each program, the initiative from BIOSES RIG, we managed to publish articles presented at the seminars, as well as issues in Scopus Index journals. So again, towards the third BIOSES 2020-21, we believe we're going to have special issue as well for the Scopus Index journals and 2 other publications. The theme chosen for this seminar is Facing Challenges Towards Advancement in Biosecurity and Sustainability, where the main focus is on topics related to many challenges, especially in biological securities and sustainability, while we are globally facing many challenges with the COVID-19 pandemic. With the 3 sub-themes, this seminar aptly fits into the Sustainable Development Goals, SDGs, very well. The sub-theme food addresses goal number 2, zero hunger. Sub-theme health addresses goal number 3, good health and well-being. While sub-theme environment addresses SDGs such as goals number 6, 7, 11, 12, 14, and 15, and we are aware that there are 17 goals altogether. Lastly, with the SDG 17 for the partnerships. Again, this seminar means partnership. It is a good platform for participants to enhance their knowledge and research skills, as well as creating new collaborations and linkages in order to become main players and future talents and the current talents in their respective fields. Ladies and gentlemen, this seminar gathers experts in the country, our counterparts from Philippines, China, India, France, and Indonesia as well. Also experts, collaborating agencies to discuss further and share their findings. Further on, to establish research-related biological security and sustainability for the future of mankind. We are pleased to have 4 keynote speakers this year. The first one being Professor Dr. Daralyn Dalise from the University of San Agustin of Philippines, who have collaborated with us for many years, already in many marine natural product studies. She's from the Institute of Marine Biotechnology, IMB of UMT, as well as the Faculty of Science Marine, FSSM of UMT. So with this partnership, we also have Professor Dr. Haji Chen from Tianjin University of China. Professor Chen is an expert in natural product and food chemistry, and she is also the recipient of the Youth Science and Technology Innovation Leader of Tianjin City. Tianjin University also has extensive collaborative work with us at UMT. We are the High Center of Excellence, the Institute of Oceanography and Environment, INOS. Professor Dr. Asadullah Khan from Aligarh Muslim University of India. He is our visiting professor at the faculty, an eminent researcher in the field of antimicrobial resistance. It was very unfortunate that he could not join us at the very last minute for BIOSES 2019, but managed to join us this time, thanks to the online technology. So it's also a blessing. We can do it virtually, Prof. To our Professor Dr. Nur Aznawati Ismail, who is from the Institute of Marine Biotechnology, UMT, an expert in the study of an archaeonic species, horseshoe crab, or locally known as blancas. And she is one of the key players in UMT Biosecurity Committee. With those, I'd like to also mention other invited speakers from multidisciplinary field, but very related and significant in biological security and sustainability. Professor Dr. Vincent Vigneron from Every University of France, an expert in artificial intelligence, AI, and bioinformatics. He has collaborated with us in 2018 and 2019, where we managed to secure the Partnership Hibiscus or PAC Hibiscus grant under My Pair program. We also have Dr. Aieni Kusrini from Universitas Indonesia, an expert in rare earth elements chemistry. She was actually our former UMT lecturer a few years back, but upon leaving UMT, still maintains good collaboration with us. Thank you. Dr. K. Soundarajan from Kogunadu Arts and Science College of India is also our collaborator from the Department of Botany and expert in phytomedicine. Dr. Rukia Hassan from Heritage Environments in Berhad, Malaysia, our industrial partner who has worked together with UMT to promote organic biocomposting as well as educating UMT students to become green ambassadors. Dr. Muhammad Anwar Jonet from Malaysia Genome Institute, MGI. MGI has an official research collaboration with BIOSES researchers from 2019 to today, which includes sharing of equipment, lab space, and student facilities at MGI. And lastly, we have our Associate Professor, Dr. Wan Iriani Wan Ismail from the Faculty of Science Marine, She is our Laurea UNESCO Fellowship for Women in Science Award recipient in 2019. Again, ladies and gentlemen, from the universities, from our local industry partner, government agency, as well as our very own expert at the university. To all the presenters who are both experienced and young researchers, your contribution in this seminar is highly valued and appreciated. Thank you for bringing your expertise, research, and innovative insights to our seminar. Your sharing will surely trigger lively discussion towards developing new research ideas and projects, networking and linkages for the improvement and enhancement towards advancement of biological security and sustainability. With 4 keynote speakers, 6 invited speakers, and 38 oral presenters, as well as 10 e-poster presenters. I'm certain this third BIOSES will be very interesting, that provides a lively virtual interaction among scientists and researchers. Ladies and gentlemen, I would like to take this opportunity to applaud the organizing committee of BIOSES 2021 for their efforts in gathering all researchers, speakers together in this seminar. To the Faculty of Science and Marine Environment, thank you for spearheading BIOSES and all other 7 RIGs in the faculty to be involved in uplifting activities such as this seminar, which is very important in spurring creative talents and research expertise in UMT, despite challenges in organizing this event. Okay, congratulations, Dr. Mariam, Dr. Erin, and the rest of the team. As for the delegates, I wish you all an enjoyable and fruitful deliberations at this seminar. Whilst at this 2-day seminar, do share your thoughts, ideas, and innovations and contribute actively to the discussion. I'm sure Professor Dato Asma will leave behind a profound message as the leader and also officiator of this event. I thank you again, Prof. Dato Asma. Wishing you all the very best. Let's work together towards a sustainable future. Thank you and enjoy your time with us. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dato, for your words. Now the ceremony wish to invite Professor Datuk Dr. Asma Ismail, President, Academy of Sciences Malaysia, to say a few words and to inaugurate BIOSES 2021. With that, please welcome Professor Datuk Dr. Asma Ismail.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera and a very good morning to all of you. Thank you to the MC and salute to my friend and sister, Professor Datuk Dr. Aieni, Vice-Chancellor of University Malaysia Terengganu, Professor Madya, Dr. Mariam Arifin, Dean, Faculty of Science and Marine and Environment, Professor Madya, Dr. Mariam Taib, the Chairperson of BIOSES 2021, respected keynote and invited speakers, honorable participants and guests, sponsors and media, the organizing committee. Ladies and gentlemen, first of all, let me wish all of you a happy and blessed 2021 with the hope that it will be a better year for all of us. We have learned a lot from COVID-19, so let us bounce back by addressing the gaps and welcome 2021 with positive thinking. I would also like to take this opportunity to thank the organizers for giving the honor to Academy of Sciences Malaysia to officiate the opening of the third seminar on biological security and sustainability, or BIOSES 2021. Now, as the Academy's president, this is an invitation that I gladly accepted in the hope of visiting UMT physically and with the enticement of nasi dagang by the organizers.
Too bad.
With the current movement control order in Penang as well as in Malaysia, that is a rain check that I will have to collect later. But I must congratulate the Biological Security and Sustainability Research Interest Group, or BIOSES, RIG, at UMT, for organizing BIOSES 2021. With the advent of the world population that will hit 10 billion by 2050, if we don't do anything about it, we will face world hunger, extreme weather conditions, livelihood crisis with the loss of jobs, and more health crises if these issues are not addressed in a sustainable and ethical manner. Hence, this conference is a timely move and a good start for the year 2021, providing the platform, as Professor Noaini has said, for many researchers to share their latest work and knowledge in facing global challenges and issues on food, health, and the environment. Ladies and gentlemen, biosecurity is a holistic concept of direct relevance not only to the sustainability of agriculture, but also to the wide-ranging aspects of public health and protection of the environment, including biological diversity. Now, as the year 2020 has been a difficult and trying year for all of us due to the COVID-19 pandemic, the theme chosen for this seminar, which is facing challenges towards advancement and biosecurity and sustainability, reflects the determination and perseverance of scientists and researchers worldwide to continue to provide solutions to these challenges. Now, as I see it, while politicians close the borders of each country, researchers basically have broken all walls by showing enhanced collaboration and sharing of knowledge in the global race to develop vaccines, diagnostics. And this is also the time where, as Professor Noaini had said, UMT had established partnerships, collaborators, and this is the time now to work together to provide impact-driven research. And I think this is the way forward to move now the high-tech industries of the country and enhance the quality of life of humankind. Now, this feeling of shared responsibility, this feeling of unity, I think will now be the strength needed for us to overcome any challenges ahead, and there will going to be many. Now, as we move now towards innovation-based economy, impact-driven research is basically the way forward to move these high-tech industries and enhance the quality of life of the people. Now, knowledge pertaining to areas of research to be discussed in this seminar covers a wide range of topics, including health, food security, and environmental sustainability. And I hope that with the congregation of experts from both public and private universities, with the industry, the government agencies, both locally and internationally, China, Philippines, India, Indonesia, as mentioned previously, I hope that this seminar will now enable participants to view the direction of biological security and sustainability, especially in this part of the world. And I sincerely hope that this seminar will be able to engage participants from the different backgrounds to share different views in research and their approaches to research, and to discuss innovative theories, methodologies, frameworks, tools, and applications in biology. That, in fact, is the plus part of digital era, whereby now democratization of knowledge is now happening big time. So with these advances in technology, let us now get together in this virtual space to take biological research to a higher level, therefore exploring uncharted frontiers in many ways that will now improve the quality of life and help provide solutions to the 17 goals of SDG. Now, ladies and gentlemen, I also hope that cooperation between universities, public universities and private universities, and with the universities with the industry does not stop at just having a conference. It should be a way forward for all of us. And that more research collaboration and networking opportunities need to come now on board, because tomorrow, ladies and gentlemen, is about collaboration. Tomorrow is about establishing of network. Tomorrow is about the sharing of information. So I sincerely hope that the knowledge gained from this seminar will be a starting point to explore potentials in biosecurity and sustainability research that will benefit humankind and therefore provide top quality scientific content and invaluable networking opportunities for researchers from many parts of the world. And I would definitely like to congratulate our Vice-Chancellor, Datuk Dr. Aieni. There are a few women Vice-Chancellors left, so we hope that more will now come on board in Malaysia and continue to break the glass ceiling. And of course, congratulations to the management of UMT and also for the organizing committee of BIOSES for doing this job and making and continuing support for this kind of seminar activities. Now, I would like to convey also my appreciation to BIOSES 2021 Organizing Committee for your hard work in making and making sure this event takes place and event taking place successfully. So let me wish you once again a successful virtual seminar, and it's hoped that the seminar will inspire the participants to contribute to the betterment of biological security and sustainability in their own capacity and capability. So lastly, with the lafaz Bismillahirrahmanirrahim, I hereby declare the opening of the third seminar on biological security and sustainability, or BIOSES 2021. My prayers is that this seminar will now become an interesting and fruitful event for all participants, and I definitely look forward to the deliverables. So stay safe and stay healthy, everyone. Thank you very much. Wabillahi taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Dato Dr. Asma Ismail, for your words. So now, ladies and gentlemen, we are extremely proud to present a montage for today's event. The ceremony which to invite Datuk Vice-Chancellor and also Dato Asma to join others for the montage presentation prepared by the Secretariat. To all participants, Professor Datuk Dr. Aieni Haji Mokhtar and Professor Datuk Dr. Asma Ismail have to leave the session earlier for other scheduled event. So therefore, the Secretariat will take a photo from this WebEx session, and then all participants and guests, please make sure that you stay cool, stay sweet, and then smile. Now, ladies and gentlemen, we would like to call upon our first keynote speaker for today, Professor Dr. Doralyn Dalise from University of San Agustin, the Philippines. Dr. Doralyn Dalise is a licensed pharmacist, registered microbiologist and natural products chemist whose expert and research interests cover microbial, marine vertebrate and plant natural products for drug discovery. She graduated from University of San Agustin, Iloilo with a degree in pharmacy and earned her Master of Science in Biology at the University of the Philippines in the Visayas. She obtained her PhD in microbiology at University of New South Wales, Australia. Dr. Dalise received a postdoctoral fellowship to specialize on marine natural product chemistry at University of California, San Diego, USA. She was engaged in several drug discovery scientific research undertakings at University of British Columbia, Vancouver, Canada, and Washington State University, Pullman, Washington, USA. She then returned to the Philippines as Department of Science and Technology, Balik Scientist in June 2015 to establish a research program on natural products drug discovery in University of San Agustin, Iloilo City. She is a pioneering scientist to work on the biodiversity, biological activities and chemistry of marine sediment-derived actinobacteria from Philippine archipelago that could one day help to find new antibiotics and potentially address the problem of drug-resistant pathogenic bacteria. Having published numerous research papers in high-impact journals and received more than 2,500 citations, she discovered more than 20 antibiotics and anti-cancer compounds from marine organisms, with 2 of them are currently investigated for clinical trials. She has 3 US patents on compounds with antibiotic and anti-cancer activities, and also won a number of awards in recognition for her research, publication and patents. She received the 2019 National Research Council of the Philippines NRCP Achievement Award in Pharmaceutical Sciences Division, 2019 Most Outstanding Professional in the Field of Pharmacy awarded by Philippine Regulatory Commission, and the 2019 Best Mentor Award for Health Research awarded by the Philippine Council for Health Research and Development of the Department of Science and Science and Technology. She is a diplomat of the Philippine Academy for Microbiology, with her presentation entitled Philippines Marine Sediment-Derived Actinomycetes, Sustainable Sources of Antibiotics. We welcome Professor Dr. Dalise.
Everyone, thank you very much for that kind introduction. So first of all, I would like to ask, can you hear me? Can anyone? Okay, all right. So again, thank you very much and good morning, everyone. First, I would like to acknowledge the presence of Professor Dato, Dr. Mokhtar, the Vice-Chancellor of the University of Malaysia, Terengganu. To the talk also of Professor Dato Asma Ismail, the President of the Malaysia Academy of Science. Good morning. And also, I would like to thank Associate Professor Dr. Ismail She's always bugging me about giving a talk and give a keynote address on this BIOSES 2021 webinar series, and I'm very pleased to be here. And what has been known and discussed before, that the pandemic will not stop us to share our research and the new findings and discovery in our universities, and this has to be shared for everyone, and thus collaboration has to come after this webinar series. And so therefore, I'm going to share my slide. Okay, so can everyone see my slide now? I'm going to. All right. So the title of my talk is The Philippine Marine Sediment-Derived Actinomycete Susceptible Source of Antibiotics. So the reason why I am focusing on antibiotics is because of the superbugs that is all over the world. It's a global threat, and these are strains of bacteria that are resistant to severally clinically available antibiotics. If we have viruses that are spreading all over the world right now, we do have also superbugs. And the superbugs have their innate property that has what they call as an antimicrobial resistance, because they are no longer killed by clinically available antibiotics. And so therefore, this AMR, what it causes today is worldwide, there are about 700,000 deaths per year. And in Europe alone, there are 33,000 deaths that was observed. In India, there are about 56,000 newborn deaths per year. And this is very alarming. And in fact, in Brazil, Russia, and Indonesia, these are all the statistics that have been shown that impact the livelihood and also the economy of the country because of this antimicrobial resistance. And by 2050, it has been projected that it can cause about 10 million deaths per year. So unlike COVID-19 that we didn't know that it is coming, the antimicrobial resistance has been projected that in 2050, it will just balloon out. And because of this, for every 3 seconds will about 100 tons in loss of production between 2016 and 2050 is being projected. And the two most affected regions are Africa and also in Asia, where Philippines and Malaysia and the neighboring countries will have the antimicrobial resistance. And so it's good that this has been projected, and so therefore we can prepare for this antimicrobial resistance. And what are the causes of antimicrobial resistance? Of course, these have so many reasons. And what particularly that is the focus of the antibiotic resistance that I am interested in is the lack of new antibiotics being developed. And this is where I am addressing my research in the Center for Chemical Biology and Biotechnology. But before moving on, I just wanted to share to you, why do we need antibiotics in the era of COVID-19? We thought that COVID-19 is just a viral infection, but in fact, there are some secondary infections brought about by COVID-19 infection, particularly with the secondary bacterial pneumonia. When the immune system is weakened in the COVID-19, there are so many opportunistic microbial pathogens that can invade the body. And this includes bacteria, viruses, other viruses than COVID-19, and also fungi. So the secondary bacterial pneumonia can follow the initial phase of viral respiratory infection or even occur during the recovery phase. And this, because of this, it increases the risk of the severity in humans. And we thought that, well, okay, COVID-19 is the one that that is causing the death, but it is a concerted efforts of all these pathogens, plus the fact that the immune system is also compromised. So this is where antibiotics come in the treatment of the COVID-19. And so if you will see here, so we have here the age of the individual with COVID-19 from 0 to 100. And most of the participants belong to 0 to 100. And so this is where the normal immunity that we have. We have the lower normal for children. We have the mid normal for most of us, and the upper normal from 60 and above. And of course, these are the hyperactivity, activated immune response, to which more than like 100 years old and above can experience. But if you look into this, even in a normal immunity, the green arrow here, you will see that bacterial complications can come in. And so therefore, the importance of antibiotics is also highlighted during COVID-19 pandemic. And there are so many evidence that secondary bacterial infection is consequential to COVID-19. In fact, these are different manuscripts that and papers that have been published recently that bacterial and fungal co-infection occur in COVID-19 patients. And if you look into the distribution of respiratory pathogens with COVID-19 co-infection, you will see on here that there are so many bacterial infections. For example, you have here the green one that I highlighted, Streptococcus, Klebsiella pneumoniae. These are pathogens that if they cannot be treated, then will cause detrimental infection to the individual. Unlike this, aside from this group of bacteria, we also have several viruses that are not COVID-19 that can have contribute to the co-infection during the process or the infection of COVID-19. And of course, we have different fungus that can play also in the infection during COVID-19 infection. And so therefore, as I was saying, the main problem that we are addressing right now is the lack of antibiotic. The antibiotic discovery in the pipeline is drying up. As you will see here in 1940s to 1970s, this is the golden era of antibiotic discovery. This is because new chemical entity are produced every year to fight against bacterial infections. But if you look at the years until now, the discovery and the development of antibiotics is drying. It is because, number one, pharmaceutical companies are no longer giving their investments in antibiotic drug discovery because the return of investment is small compared to investing in drug development that can cure lifestyle diseases. And so therefore, they disregard antibiotic drug discovery. And now what picks up the antibiotic drug discovery are the small companies, and particularly the academic research institutions. And so this is where we as a university could play in with and contribute in the discovery of new antibiotics. So where can we get most of them? So we get most of them from actinomycetes. However, there are also coming from other bacteria, other gram-positive bacteria. We also have the fungal metabolites, the penicillins, and then we have the synthetics that have either, you have the fluorine, they are halogenated, and you have the azoles and the quinolones and the sulfonamides. And so most of which of the antibiotics are taken from actinomycetes. And these are examples of antibiotics that were discovered from actinomycetes. So actinomycetes is under an actinobacteria, and it is a soil a soil bacteria. If you grow them in a petri dish, what you will be seeing are this beautiful colony that you thought these are fungus, but it is actually a bacteria. They have mycelium. They have also spores. As you can see, they have mycelium here, and they also produce different spores. I consider them as the best chemists in the whole world. It is because they produce tons and tons of chemical factory based on whatever stress they could encounter, whether stress in brought about by competition in food, competition in nutrients, competition in space, or competition in light. And this is something that we could exploit in finding these two different antibiotics. So these are examples of antibiotics coming from the soil bacteria, and most of you are familiar about this. You have the vancomycin. These are very expensive antibiotics now. We have a tetracycline, but not totally used by many because of its side effects. You have also rifampicin or rifamycin. We have amphotericin B, streptomycin for Mycobacterium tuberculosis, and erythromycin, which is very close to my heart, is because erythromycin is a metabolite of streptomyces that was discovered in my city called Iloilo City by a pharma company by a pharma company wherein they collected a soil from my town and brought it to their lab and found this erythromycin, erythrus producing this erythromycin. And so in honor of the city where they took the soil, they named erythromycin with a brand name Ilozone to honor Iloilo for its origin of the Streptomyces erythrus. And so therefore we are reviving that kind of discovery, but this time all the intellectual property will be made by the Filipino scientists for the Filipino people. And so here, this is a diagram of how metabolites are produced. Of course, we know that these are the primary metabolism. These are metabolites used by bacteria in order for them to grow, essential for growth and reproduction. But if you look at the secondary metabolism, these are more complex. These are primary metabolites that have been biosynthesized to make elaborative chemical entity so that it could be used in other processes aside from growth and reproduction in bacteria. And so this is where all these antibiotics produced by actinomycetes originate, from this elaborative secondary metabolism brought about by polyketides and phenylpropanoids by synthetic pathway. And so if you look at this, the polyketide synthase is the chemical biosynthetic gene clusters that creates all these amazing metabolites that has bioactivity. And if you look at this, they are very diverse. Unlike plants, plant metabolites are not as diverse as bacterial metabolites. It is because they do not have a very big biosynthetic clusters we call as PKS. But the polyketide synthase, you will see that there are different metabolites. You can see a small ring, a big ring, and they're more elaborative. And so therefore, they could target different types of enzymes that we could exploit either for industry, either for health purposes, and other types of applications that are beneficial to us. So going to where we are as a collaborator, we are collaborating because we have one thing, and our theme is we're working on the marine ecosystem, and in some different forms. So we took this video when we went to the Tubataha National Reef. This is one of the biggest reef in our nation, and it is considered as the UNESCO National Heritage. So the marine ecosystem, you will see a lot of biodiversity. Each of the individual organisms there, through evolution, was able to develop strategies that they can defend themselves. And through these evolutionary processes, they were able to produce these different types of compounds that are specifically targeting their prey, either fish, it could either be mollusk, it could either be other predators in the ecosystem in competition for nutrients. And this metabolite is something that we could exploit in order that we're able to get what we call as a compound that can help us to alleviate diseases in other forms. And so here in University of San Agustin, we are doing a natural products drug discovery program, working on these different types of natural resources. We have marine invertebrates, plants, and also marine microbiology. I'm not talking on the marine invertebrates in my keynote address right now, and also with plants, but I will talk more on the marine microbiology. So in marine microbiology as a source for natural products, we have all these different platforms that we are creating in the center, from isolating pure forms of bacteria until looking into producing extract library. We have thousands of extract library in our center. We also have our in-house in vitro assays and different antibiotic target sites. We also have anti-cancer. We do a lot of isolation purification, but in our isolation and purification, we do, prior to isolating that compound, we do metabolomics work to identify or to trace what is best. And then we also do in vitro evaluation, because we have our mantra here in the lab that we would rather fail early than fail later. We want to make sure that the natural product that we are going to develop into a drug is safe. And so therefore, in vitro evaluation into cytotoxicity against liver, kidney, and lung cells are done in this lab to make sure that these are all safe. We also have genomics to mine the genome of the organisms that we isolated. And from there, all of this, we have drug leads for drug discovery. So I have my collaborator, Dr. Junel Saludes. Both of us are actually natural products chemists, but my background is more in biology. Dr. Saludes is more in chemistry. And so therefore, later on, when we have the product or the natural product identified, then we were able to do a lot of tweaking on the structure to identify, to make it more effective, less toxic, to have a potential drug lead. And so this is the map of the Philippines. The green dots here were the collection site that we have done in the last 2 years in looking at the sediments of the Philippine archipelago. So this is our strategic approach. We have all these different approaches. It doesn't mean that it is in order, but this is usually what we've been doing. We collect marine sediments, do a pure collection, selective. We have also come up with a selective marine medium that what we could get are actinobacteria and nothing else. And so that is already been filed for patent. We also have a fermentation and secondary metabolite extraction, bioactivity against escape pathogens, because we are interested, the escape pathogens are more drug resistance than any other pathogens out there. We also have the anti-cancer screening, and the bulk of our work in chemistry is the purification and isolation. We have the whole suite of this. We have also metabolomics, LCMS, and chemical profiling. So the center has LC, GC, MALDI, mass spectrometry, and also QTOF mass spectroscopy to identify the metabolomics. We are doing 16S sequencing and other amplification of different types of enzymes involving the polyketide synthase, and then we identify the bioactive compounds. We also have been doing MIC determination, IC50, and most of our work is also mechanism of action studies, and as again, the toxicity assay against normal cells. And so therefore, I'm going to bring you to our university. So our university is a very small university. It's a private Catholic university. It's in the heart of Iloilo City. This is our center here. We call Center for Chemical Biology and Biotechnology. So this is again the map of the Philippines. Our area is here. And then I'm going to bring you to our expedition sites in the Tubataha Reef in the Palawan. So we do not have our own vessel because we are just starting. And so we have our collaboration with the MY Palau Sport when we were doing this. So this is our team. I'm the only female in our team together with the different divers. What we have is a sediment core sampler that we lower in the seafloor, and we fabricated this back in the States. What we're doing, our robotic system, and really the hammering is through gravity because it's very heavy. But here in the Philippines, we have to do it manually. So the 1 meter core has to go at least 1 meter deep in the seafloor. And we are interested to look at the different types of actinomycetes in different layers from the surface of the seafloor down to 1 meter of the area. And so for the first year in 2017 and 2018, we were concentrating our isolation and expedition in the central of the Philippines. So we have all these different types of areas and pristine environment away from the islands where most of the people are living. And so therefore, this is one of the areas that we looked into. And in year one, we were able to collect at least 11 sites and collected at least about 25 core samplers in this area. We are only doing this in the month of March to June because this is where we have no typhoon, because outside that month there is heavy wind and typhoon season. So we have only that period, a 3-month period to cover our expedition. And in year two, we went beyond the area where we have in year three. So we went to different areas, even up north near Taiwan. In Batanes area, we're able to collect some of this. And then we also went to the eastern part of the Philippines and one facing the Pacific Ocean here in Lahanosa Island, and also down south, where we're able to get some of our samples in the area. So pretty much all of this have been covered. And so therefore, what I'm going to show you here now is in the last 2 years, we were able to isolate 3,000 pure isolates of actinomycetes. It is a big library, and I was even amazed to see how diverse they are based on their diffusible pigments that we're able to see. I've been working with actinomycetes that were recovered in Bahamas area and also in the Pacific, North Pacific from California and South Pacific in North California up to the Vancouver area of British Columbia, Canada. But I haven't seen such diversity of these actinobacteria than what we have seen right now. So we consider this as a drug discovery biobank in the Philippines. This is not only an antibiotic source, a biobank, but we could get a lot of small molecules here that has the application for industry and health purposes. This I consider is a sustainable source for research. Unlike marine invertebrates, I've been working with marine invertebrates for the last 8 years. And what I've been seeing is that, yes, they produce these amazing metabolites that we could use for health and industry purposes, but in the long run, if we harvest them, there will be no one left in the ocean. It could either be that they will be vanished because of over harvesting and not replenishing them. And so therefore I said I will focus on something that is sustainable, meaning I can get it from a marine ecosystem and then provide nutrition and grow them in the lab. That anytime I would like to say, okay, I would like to investigate this particular actinobacteria, what I would like to do is not to go back to the ocean, but go to the -80 freezer, find that in the biobank library and grow them according to what my application would be. So this is how sustainability works in our research center. We are not going back to the marine ecosystem to disturb again the marine sanctuary, because we already have this wealth in our system. And in fact, the sediments that we have, it is still in -80 freezer. And what we are doing in that is we are still working on it because we have found some other unique organism, novel organism that is related to streptomyces that we could exploit to find more bioactive compounds. And so what we do is we do a mining approaches in the natural products discovery. We have our metabolomics, we have our genomics, and in vitro bioassays. So these are very independent. So there's a group working on metabolomics, there's a group working in genomics, and there's a group working the in vitro bioassays. But it comes in a collaboration, and the main purpose is to find a new and a bioactive metabolite that sometimes we could get metabolites that are not new. However, the bioactivity is new and there is novelty into that. And so with the genomics, we do of course the usual phylogenetic markers, the 16S, but we're looking into this protein coding genes, because you will be surprised that, yes, they are very, very identical in their 16S RNA, but their protein coding genes are not, they are not identical. And so we have one paper that is coming up to describe different types of multiple strains from a particular bacteria and also its antibiotics that they are producing. So we are also interested on the small metabolite biosynthetic gene markers. So we have the PKS here, 1 and 2, polykeptide synthase, and the non-ribosomal peptide synthase. And through this, we could look into the phylogenetic tree construction and even find something interesting there. And so again, this is the escape pathogens that we look into. We have the escape, as you can see, the initial is called escape, or they call this is escape, and they have its corresponding scientific name. And these are multi-drug resistant, they are virulent, and they can cause respiratory disease. And during COVID-19, these are the group of bacteria that can cause opportunistic infections during COVID-19. So one of our papers were published earlier in April. This is in Frontiers in Microbiology. We were able to discover an anthracycline shunt metabolites for the first time in the literature, because these shunt metabolites have not been isolated in a bulk quantity, because they are used up immediately in the synthesis of anti-cancer compounds. And you can only get and isolate anthracycline shunt metabolites when you have a genomic modification of a bacteria. But what we've seen is from the wild type, we're able to find these anthracycline shunt metabolites. So these are the group of people that work on this: Melissa Padero, Edna Sabido, and Angelica Suarez. We were able to get the sediments from this small, tiny island we called Lubong and Alad Island in the Romblon group of islands, and we were able to get this bacteria called Streptomyces griseorobens. And of course, we have a library, and what we found is we are interested on this isolate. And I said, okay, look into that and then see what we can find. But on that, we've seen that with 16S, the compound and the bacteria DSD069 is very related to Streptomyces griseorobens. And so therefore, we call this as a Streptomyces griseorobens strain DSD069. So this is how it looks like. They are not that filamentous, but they have a lot of these spores, and their spores are has a size of about 1 to 1.5 micron, and you will see this is from the plate, and then with the electron microscope you will see all these tiny amazing spores that this is actually the one that we get from the marine sediments. It is the spores that we get from the marine sediments, and we get the spores back to the lab and then grow them to these amazing actinobacteria organisms. So what we've seen, this is the time kill assay that we have found. This is the antibiotic kinetics, and we've seen that the griseorobens is comparable with tetracycline. So you will see here that at 6 hours, it is capable of killing almost 60% of the population. But of course, the tetracycline, after 6 hours, almost 99% of this are killed. But you will see that the antibiotics produced by this isolate is killing it until 12 hours, and eventually 24 hours. The SRUs is totally killed, just like the tetracycline. And this is something that we are still working on the kinetics of its killing, the killing kinetics of the antibiotics. And so one thing that we're interested is if it kills the bacteria based on that graph or that curve, how do we know, or how does it kill the bacteria? So first of all, we are interested to look into whether the protein or the DNA are affected. And so first we look into if it affects the protein and the DNA, it has to have an effect on the cell membrane. And so therefore, we look into the amount of the protein and the DNA that were released after the treatment of the antibiotic. And so we measure the protein and the DNA. And you will see that compared to the twin AT that lyses the cell, you will see that at different treatments of the antibiotic, you will see that there is the amount of DNA that is released, and also the protein. So I said, I'm not quite convinced on this. And so therefore, we have to look into the structure of the cells after it is treated with antibiotics. And so we look into the transmission electron microscope. And we have found that the untreated and the DMSO, as we consider it as the negative control, you will see that the cells of Streptomyces are very round, they're happy, they are growing well. But with the DSD069, with that antibiotics, you will see that the cells clumped together, they are collapsed. Their cell integrity is gone. And so this, I said, wow, this is something that tells us that when they collapse, the proteins and the DNA are released out of the cells. That's where we were able to fish out and measure how much of the protein and the DNA were released. Moving on, I said, well, I have to see more of convincing, supporting evidence that indeed it is targeting the membrane or the cell membrane permeability. And so we look into the flow cytometry. So we dye the cell with a propidium iodide and calcine. The propidium iodide can penetrate the cell membrane, only penetrate the cell and intercalate in the DNA only if the cell membrane is compromised. The calcine cannot go into the cell because this is a probe that can only dye metabolically active cells. So looking at this, we have two types of population. We have the R2 and the R1. R2 populations, these are the cells that are very happy. These are cells that are with fluorescent calcine, so they appear green. But on the R1 population, these are cells that are not happy. These are dead cells. So looking at this, A is the untreated, B is the DMSO. You will see that they're the population of happy and live cells. C is the ethanol that we use to kill the cells. So you will see that the population in R1, most of the cells are dead. The R1, the D graph here is actually a vancomycin. So we thought, what's happening with vancomycin? Vancomycin, after 3 hours, there are still cells that are happy. Less of the cells are dead. It is because vancomycin is not a cell membrane permeability target or antibiotic, but instead, its target is the cell wall biosynthesis, and it makes sense. So because the cells are not, the cells has to grow and to synthesize the cell membrane, and so therefore the cells still here are very happy. But in E, you will see that there is a big. This is treated with the antibiotics. These are the cells that are not happy anymore. So these are the dead cells. And then these are the some cells that are still happy. So these were captured after 2 hours of incubation with the antibiotics. So what are the antibiotics that we're able to get? We have the shunt metabolite. You have these 3 contiguous rings that are highly UV fluorescent. When we look at this in the UV profile in our HPLC, you will see a very big peak of the UV 254, and we're able to get this bis anhydrooclavinone and 1-hydro bis anhydrooclavinone. And we've seen that this has a low MIC than this. And we suspect that if you're going to add functional group here, then it will affect its antibiotic activity. It will no longer be as effective as it should be. So this one, it could be that there is a steric hindrance brought about by the hydroxyl group here in the C1 position. And so therefore, this is also a good way of showing that structure and activity has a relationship. And so therefore, these 2 anthracycline shunt metabolites has a novelty in this paper. So we also provided a putative biosynthetic pathway because generally what have been shown is that you cannot fish out all this too, because all of this will be turned into anti-cancer compounds, doxorubicin and also daunorubicin. But with the DSD-069 isolate plus our medium and the fermentation mechanism or processes that we have, this aclavinone were spontaneously formed into bis anhydrooclavinone, and then we asked this question: were we able to find doxorubicin in the extract of DSD-069? And we weren't able to see that. And what we've seen as a major molecule or major metabolite are these shunt metabolites, because instead of going down here all this biosynthetic pathway goes to the left to form these two anthracycline antibiotics. Another paper that we recently published in Journal of Marine Science and Engineering, in a special issue, Diversity and Biotechnological Potential of Marine Microorganism, is this organism that we got just about 4 hours from where we are located. It's called the Islas de Gigantes. You were able to get this organism we called Streptomyces sp strain DSD011. It has filaments and it has a spore that is about 0.5 micron. So when we were looking at the 16S, it is similar to cellulose, but you will see that the bootstrap value here is very low. And so therefore I said, let's go to the protein, to the housekeeping genes, and we will see what's going on. And so with the concatenated gene sequence of RPOB, ATPD, and other housekeeping genes, we're able to come up of a phylogenetic tree, and we found that what we've seen is actually novel from the rest that where it is similar, and this is a novel thing that we were able to identify. And this type of streptomyces, these were reported from the terrestrial origin, but they were isolated from the hypersaline environment in the terrestrial in a terrestrial site where they are located. So we hypothesized that our strain is a streptomyces from the terrestrial environment that has been washed off to the ocean, and in the ocean it develops its own evolutionary adaptation with the marine environment, and so therefore it diverges from its other relatives in this phylogenetic tree. And we've seen that this is salt tolerant. So it can grow at 0% sodium chloride, 3%, 5%, and even 7%. And again, this could be a relative of those in the phylogenetic tree, because these are from the hypersaline terrestrial environment. And so we've seen that this is a salt tolerant, a new species of a salt tolerant streptomyces species that have been washed off into the marine environment. So we looked into the PKS and the NRPS, and we found that what it has are these PKS genes. We're able to amplify it more. But with the PKS1, they don't have that much. Well, I would say that they do not have a PKS1. It could be that the primer that we use is not very specific to the PKS1 present in the genome of the organism. But what we've seen is that 3 of the PKS2 amplified gene, we were able to see that, wow, this is something that we have to find. What is this PKS2 compound that they are produced? With it also has the NRPS. And what we've seen are actually a PKS2 type of molecules. We call that as angocycline glycosides. So because of the presence of the sugar attached in here, and we call this as freedomycin A and freedomycin D. What we've seen here again is the structure-activity relationship. You will see that all of them, both of them, has this chemical structure. Seen here, they are identical, but the sugar molecule here, they are not identical. This has only one ring, one sugar ring. This has 3 sugar rings. And if you look at the MIC, it has 62.5 microgram per mil compared to this that has 500 microgram per mil, indicating that the addition of this 2 more rings in this moiety has added to the increased antibiotic activity. So moving on, we have some other types of organisms that we work in the lab that we are, the manuscript that we are actually preparing for publication. This is DSD-42. It is available or very similar to Viridio diastaticus, but actually it is again a novel compound compared to the rest of, not a novel compound, a new species of Streptomyces strain compared to its relatives here. So what is good in here is we're able to identify that these are all from, they target escape pathogens, and we are interested in Acinetobacter baumannii here. So after gel filtration or gel formation chromatography, we're able to get different types of fractions. You will see that the crude will now have an increased activity. And what we have right now is a very promising antibiotic that we've seen as after gel filtration, we purify this further in flash chromatography. You will see that it has a bigger zone of inhibition compared to tetracycline, and both against these gram-negative bacteria. And so I am not, and we've done a HPLC on this, and I cannot disclose the structure of this compound, and I will just share to you the manuscript once it is published. So moving on, we have the diversity and abundance of this in the Streptomyces. I'm going to show you the whole picture of what we've got. A paper will be coming out soon about this project. And so we have about 2,780 pure isolates that we tested against the escape pathogens, and we have 134 of this that are antibiotic producing organism. Most of its targets SRUs, a little have the Pseudomonas aeruginosa, and then we have the E. coli, and then we have the Enterococcus aeruginans. So this is a very beautiful slide showing the relationship of the 16S RNA with the functional traits, meaning its bioactivity, both the anti-cancer and antibacterial activity, and also polyketide synthase and the NRPS. So you will see that each of this color is a one species. One species of Streptomyces. So this is Streptomyces parvulus, and this is a Streptomyces inocenensis. And you will see that we have also a bulk of Streptomyces rochei. But if you look at each of this, so if I'm going to focus on Streptomyces parvulus, these are all identical strains of Streptomyces parvulus. But if you look at their functional traits, meaning their antibacterial activity, you will see some have activity with SRUs, some they don't. Some have activity with E. coli, some don't. Some have activity with Pseudomonas aeruginosa, some don't. And this is an exciting scientific findings that we have seen. And so therefore, we are mining the genome right now of these different types of multiple strains of bacteria to find what makes this strain different from the other strains, although they are entirely identical. And so here we have the distribution abundance of actinobacteria from different sampling sites in the Philippines. And you will see that some have its high diversity, some don't. Example, zero diversity in Central Visayas and North Camigan because you only got one type of Streptomyces. But if you look at the two Bataha reefs, the one that is very far from the other islands in the Philippines, we have about 2% diverse when it comes to different types of Streptomyces species. We devise our own culture-dependent approaches. We have We are not following the traditional way of culturing actinobacteria, and so therefore we modify and devise our own. And so we call this the heat shock method and the dry stamp method. Once the paper is out, you will understand, or you will read more closely what's the difference between the heat shock and then the dry stamp method. And we were able to see that if we use the dry stamp method, we have higher diversity compared to the heat shock method. Then we have also used different types of carbon sources. So we devised our own isolation culture media, and we filed a patent on this. And we found that if you use different types of carbon sources, it will affect the diversity of the streptomyces that you can get from the marine sediment. Like, for example, raffinose, this is not used in a traditional isolation of streptomyces, but you've seen that there is about 2%. The tree halos is something that is new to most actinobacteria community, but we use here it in our case, and you were able to get about 2.3. The most popular media that is used to isolate streptomyces is the starch, but see that the diversity is very low. So we were able to identify carbon sources and a media that were able to get a good diversity of streptomyces compared to the traditional starch that most of the streptomyces group have been using for such a long time. This is another amazing, another scientific discovery that we found in our paper. Remember when we have that one core sampler that we hammer from the seafloor until 1 meter on that seafloor. So the seafloor, so we have 1 meter of the core sampler. So we divided it in different depth. So we considered it is from the bottom, sub-bottom, middle, surface, and subsurface and surface. And we've seen that diversity index is gone to about 2.3 if we go below 1 meter from the seafloor. Imagine if you have 5 meter of that core sampler. Imagine that if you will isolate actinobacteria there, you will have you expect a higher diversity. And so you will see that these are the distributions of the active antibiotic producing streptomyces strains that we have found, and we have here the corresponding streptomyces. So we've seen that there are those that, for example, in this area here, in the subsurface, and then we have the bottom area. These are the specific organisms that we have found that are not found in the surface. And so you will see that in the surface of the seafloor, you have parvulus. S. parvulus is a ubiquitous terrestrial organism. So we hypothesize that these S. parvulus are the new spores that have been washed out from the terrestrial environment to the ocean and was able to occupy the surface of the seafloor. And so we've got a lot of this S. parvulus. And there is more into that in the discussion in our paper, and I'm going to share that to you once it is published. And so here it is. I was telling the S. parvulus, we have so many of this. They are very identical with their 16S and their housekeeping genes. But if you look at their functional traits, they are not identical. So look at this. So these are the group of Streptomyces parvulus that are only active in E. coli. And also, these are the two Streptomyces parvulus that are active only in Pseudomonas aeruginosa. We also have Streptomyces parvulus that are active to 3. And so this now poses a question. are the metabolites produced by this multiple strains of S. parvulus the same or not? So we are looking at the metabolomics area on this, and we are also preparing a manuscript to describe the metabolomics, the individual antibiotics or individual metabolites that are produced in this site. And so, in summary, the drug leads from the vast Philippine marine sediment-derived actinobacteria has unlimited potential to offer economic and healthcare benefits to the society to fight against infectious diseases. And I will say that it is very sustainable. And so, therefore, the Philippine marine sediments are rich in diverse actinobacteria that may serve as valuable resource for antibiotics. And I will encourage those researchers in UMT to explore the marine sediments near your area and the area where you have a jurisdiction that you could get marine isolates and sediments and bring that to the lab. So I would like to acknowledge our funding agency, the Department of Science and Technology, and also the Balik Scientist Program, PCHRD, and the National Research Council of the Philippines. And this is our team, and we are still growing. We are also inviting visiting professors to come in our lab to join us and have this collaboration. We also have our scuba tech that we partner for our marine expedition. And of course, when we do our expedition, it has to be with the agreement and the collaboration of the local government agency in different areas in the Philippines. So without them, we weren't able to get samples in their vicinity. And please follow us in Facebook, Instagram, and Twitter, and email us should you have questions, would like to have a collaboration, or visit our lab as a visiting scientist or a visiting scholar. So thank you very much for your kind attention.
So thank you, Professor Dr. Dalise, for your keynotes. Next, we are going to have Professor Dr. Haji Cheng from Tianjin University, China. Haji Cheng is a professor in the School of Pharmaceutical Science and Technology at Tianjin University. She accomplished her PhD in 2002 in Natural Products and Food Chemistry from Huangzhou Agricultural University, China, and she received nutritional and pharmaceutical research training as a postdoctoral fellow and visiting scholar from Ocean University, China in 2002 until 2004, and University of California, Berkeley in 2011 until 2012, USA. She has a broad background in the chemistry, nutrition, pharmacology, and toxicology of food, natural products, and traditional Chinese medicine. She has focused her studies on natural products for more than 20 years. She has brought forth new ideas on the investigation of bioactive natural constituent and achieved many research achievements on the isolation, preparation, and identification of the natural products, activity screening and efficacy evaluation, pharmacology and toxicology of the natural products. Her research will be helpful to the investigation and utilization of natural resources. She has outstanding records of scientific and academic accomplishment with more than 15 research funding as principal investigator, more than 100 publications in highly prestigious international journals, and various presentations in both national and international conferences. She has 5 books published, 35 Chinese patents applied, and 15 of them were authorized. Her papers on the study of bioactive constituents have been cited by many researchers around the world, and the citation frequency reached 2,200 times in SCI database. She has been the editorial board member of some journals such as EC Pharmacology and Toxicology, Journal of Food Safety and Quality, Pharmaceutical Journal of Chinese People's Liberational Army. And she also has been the reviewer of NSFC, most and many international journals and Chinese journal. She has been awarded the Youth Science and Technology Innovation Leader, the first level of 131 innovative talent of Tianjin City. With her presentation entitled Network Pharmacology Studies on the Bioactive Compounds and Action Mechanism of Natural Products for the Treatment of Diabetes Mellitus, now we present to you Professor Dr. Haji Chen.
Hi, hello everyone. I'd like to know, can you hear me? Yes. Okay, thank you so much for the kind introduction. First, I'd like to thank for University UMT for all the organizers to give me the opportunity. I'm very glad to have this opportunity to communicate with you. I'd like to thank Vice-Chancellor and all the professors and dear ladies and gentlemen. Good morning. So I'd like to share my slides. Can you see my slides?
Yes, we do.
Okay, today my talk will be focused on network pharmacology studies on the bioactive compounds and action mechanisms of natural products for the treatment of diabetes mellitus. And this is my general information. If you are interested in the natural products from the marine or from the land, and you're welcome to communicate with me. And my research interests are mainly focused on the following three directions. First one is about the structure, bioactivities, and the mechanism of the constituents from natural resources. And we mainly focused on the structure, the isolation, the elucidation of the structure, and the bioactivities, functional mechanisms, and the structure-activity relationship of the macromolecules, such as the polysaccharides and the protein, and the small molecules, such as the flavonoids, the terpenoids, and the alkaloids, etc. And then the second research direction in our research group is focused on the analysis method study of the chemical constituents. And we want to develop the new methods and employ them to qualify the bioactive polysaccharides or the proteins and the small molecules from the natural sources and the traditional Chinese medicine and the other sources. And then the final target of our research, that is to develop the final product that is a functional food of the new drug, and then that made it for the application for the human peoples to use them for the healthy effects. So we want to explore and develop the new functional food and the new drugs from the natural sources. So today I will give you the main topic is on the network pharmacology studies on the bioactive compounds and the action mechanisms of the compounds for the treatment of the DM. As we know that with the development of our living life, many people are suffered from many kinds of chronic disease, such as diabetes, and hyperlipidemia, and the hypertension. So, as we know that on the World Health Day in 2016, there is the call of the beat diabetes, that is the action needed to halt the rise of the diabetes. And in 2019, the IDF estimates that 1 in 11 adults, that is between 20 to 79 years old people, they will have diabetes. The number is about 463 million people. And 1 in 2 adults with diabetes are undiagnosed, and over 3 in 4 people with diabetes live in low and middle income countries. So there is the prevalence of the diabetes. And especially the diabetes will cause severe complications such as the kidneys, the eyes, the cardiovascular diseases, and the stroke and the function failure. So action should be needed to treat the diabetes. Let's see the map, the diabetes mellitus epidemic from the 2003 to 2025. In the world, in 2003, there is only about 189 million people suffered from diabetes. However, according to the estimates, in 2025, there will be 324 million people will suffer from diabetes. So the prevalence of diabetes is severe. And according to the number, let's have a glance. In 2019, the number of the people with diabetes, that is 463 million. However, in 2045, the number will increase to 700.2 million. So the 2019 edition of the global diabetes map, the number of the diabetic patients worldwide, among them, and China has more than 116.4 million diabetic patients. And the following, the second one is India, and then is the United States of America, and then is Pakistan, and especially many countries are suffered from this disease. There are 3 categories of the diabetic. First one is called the T1DM. That is type 1 diabetes mellitus. It is caused by the insulin deficiency related to the dysfunction of the beta cells. And the second type is called the T2DM. That is the type 2 diabetic mellitus. It is characterized by insulin resistance and relatively insulin secretion reducing. And the type 2 diabetes is the main category, account for about 19 of the diabetes cases. And the third one category is the GDM, that is the gestational diabetes mellitus. It is characterized by high blood glucose levels in pregnant women without previous diagnosed diabetes. And there are many current used oral drugs. This table you can see they have different categories and they have different action mechanisms, such as the sulfonylureas category and the biguanide category and the iphoglucosidase inhibitors categories, and the DPP4 inhibitors. So you can see based on different categories, they have different action mechanisms, and however, they have many kinds of side effects, such as the hyperglycemia, the GI symptoms. They will cause the abdominal cramping, and maybe they have the hepatotoxicity, and they can cause the headache or hypersensitivity and the skin reactions. So we need to find the new candidates for the treatment for the diabetes. So because our research is focused on the natural products, we know that now there are many kinds of natural products have played significant roles in the race for the drug discovery, which possessed approximately 64 of all the drugs. And the new drugs candidates and the functional foods for the diabetic from the natural researches has focused on the different kind of natural sources. And maybe they can focus on their studies on the extracts, and based on the compound categories, we can divide them into polysaccharides, the polyphenols, the terpenoids, and the alkaloids, and the lignans, etc. So different kind of natural sources from the land or from the machine, they all can be discovered for the candidates for the treatment of the diabetes therapy. Let's see the different characters of the traditional or the conventional therapy and the novel therapy. We can see that based on the general oral drugs now we are used, they are the conventional therapy. They just focus maybe just one target and one drug, and they may have side effects. That is the character of the synthetic drugs. However, for the natural products or the network pharmacology focused on the natural products or drug candidates, they are called the novel therapy treatment methods, and they have the character that they have multi targets, multi components, or maybe they have no, or maybe they have less side effects. So that's the privilege of the natural products and the natural network pharmacology. So let's see what's network pharmacology. We can see that it integrates the network biology and the pharmacology. It is considered as a promising approach toward more cost-effective drug development, and it emphasizes the regulation of the multiple signal pathways, improving the therapeutic effects and reducing the side effects so as to improve the success rate of the new drug clinical trials. and it coincides with the holistic and systematic concepts in traditional Chinese medicine theory, and also many other countries' traditional medicine can use this kind of method. And the TCM network pharmacology has been applied to the herbal ingredient target disease gene prediction, network balance regulation, and the elucidation of the synergistic ingredient pairs and active ingredient groups. So here you can see other examples for the TCM network pharmacology. It can be used for the herbal ingredient target or disease gene prediction, and it can be used for the network balance regulation and the herb network and the active ingredient groups, and then we can find the drug candidates on the groups. And here you can see other examples of the natural products for the DM therapy. According to the compound categories, they can be divided into many types, such as the lignans, the polyphenols, the flavonoids, and the terpenoids. All of them can be used for the treatment for diabetes. And there are many databases can be used. They are important for screening in drug discovery, which can provide the related information for the natural products for the prediction for the disease treatment. So you can see here are some examples of the databases, such as the Supernatural 2, the Naprolator, the drug bank, etc. You can see they can provide the two-dimensional structure information and the corresponding structural and the physical chemical property, and the predicted toxicity class, and the vendor, and the name, code, or country, pharmacological effects, etc. So you can see different databases. They can provide different information. So we can use this kind of databases. And there are also some databases related to the type 2 diabetes. You can see such as the type 2 diabetes DB and the type diabetes GAD, and the Zhejiang University. They have this kind of databases, so they can provide the transcription, the unigen and the homologen and the tissue-specific expression, the pathways, and the PPI-associated risk factors or complications, and also the summary of the type 2 diabetes related genetic association study results, and also some levels laid out on associated connections or basis for the further study of the type 2 diabetes in system biology, etc. So if we focus our study on the drug discovery on the type 2 diabetes, we can use this kind of databases. So the related databases, they can have the high availability of these databases plays a significant role in the related research. And these databases can be used to predict the target network of the bioactive constituents, and after experimental validation of the molecular network, they can provide a new perspective for the researchers to better understand the pathogenesis of the diabetes and the action mechanisms of the associated drugs. So let's see, when we want to discover a new kind of drug on the type 2 diabetes, maybe we need to know the related mechanism. So let's see the action mechanisms related to the type 2 diabetes. The following four main related mechanisms are the hot research area. First one is about the alpha amylase and the alpha glucosides inhibitory mechanism. And the second way is about the targeting beta cell dysfunction. And the third mechanism is focused on targeting different types of the signal pathways. And the fourth one is the modulation of the gut microbiota. So let's see the first one mechanism, the alpha amylase and the alpha glucosides inhibitory. So as we know that the dietary carbohydrates can be hydrolyzed to the oligosaccharides, the disaccharides, and the monosaccharides based on the enzyme, especially the alpha glucosides and the alpha amylase. So if we have our natural products to inhibit the activity of this kind of enzymes, we can have the activity on the treatment of the type 2 diabetes. And also the natural products may have the anti-inflammatory or antioxidant activity, so they can cause the inhibitory of the beta cell dysfunction, because the inflammation, the oxidative stress, and the overconsumption of the fat and free fatty acids, they can cause the beta cell dysfunction. So if we have the natural products candidates, they can have the anti-inflammatory and the antioxidant activities. So they can inhibit the beta cell dysfunction, and then they can have the anti-diabetic effects. In terms of the third action mechanisms, that is the targeting signal pathways. As we know, the type 2 diabetes, they have different types of the signal pathways, such as the AMPK, the PI3K, AKT, and the ROS-ERK, NF-kappa B, and the mTOR, WNT, JAK state. You can see different signal pathways can be involved in the type 2 diabetes. So if our natural products can have the regulation activity on this kind of signal pathways, then they will have the anti-diabetic effects. So here you can see is what we have summarized for the signal pathways related to the diabetes mellitus. So the main signal pathways are the about summarized. We have summarized here, such as the AKT, the mTOR, the AMPK, and also the MERK and the ERK pathway, and also the GLUT4 pathway. So when we want to discover the anti-diabetic drugs, we want our candidates can target these critical cytokines, and then they can regulate the related pathways. So here are the examples for the research. First is about the targeting signal pathways, AMPK, such as the black soybean seed coat extract. It had rich compounds of the anthocyanins and the procyanidins. And you can see here is what found the AMPK upregulates catabolic pathways that generate the ATP and downregulate anabolic pathways that consume ATP. And the black soybean seed coat extract, the BE, can significantly increase the phosphorylation of the AMPK alpha at the 172, and the phosphorylation and the total AMPK alpha ratio. So the BE can also significantly increase the glutathione protein expression and activate the AMPK, downregulated the expression of the gluconeogenic enzymes and contribute the reducing the blood glucose concentration. So that's the mechanism why the BE has the activity on the diabetes. And another example is targeting the signal pathways, the PI3K, AKT, and such as the bunksol fruit oil extract. It is rich in the palmitoleic acid. So you can see it can increase the expression of the PI3K and consisting of a catalytic subunit and regulatory subunit. And this extract can also reduce the expression of the GSK3 beta. So that's the mechanism on the SBFO have the anti-diabetic effects. And another example is on the berberine. It is a kind of alkaloid. And you can see here the results, the BBR, it can downregulate the expression of the mTOR and the phosphate mTOR by upregulating the expression of the AMPK and the phosphate AMPK. And the BBR can also enhance the autophagy and reduce the podocyte injury induced by the high glucose by regulating the mTOR signal pathway to treat the diabetic nephropathy, one of the diabetic complications. And another example is targeting the signal pathways event. The total glucosides of the pony, we called it the TGP. And the mechanism is about that the TGP can downregulate the expression of the VINT1, the beta-catenin, and improve the kidney damage in diabetic rats and delayed the development of the diabetic nephropathy. And another example is targeting the signal pathways, the JAK-STAT, and the constituent is the tryptophan refolded polysaccharides, polyglycosides. It is a kind of glycosides, and you can see it can downregulate the expression of the phosphate JAK2 and the phosphate STAT3 and ICAM1 and IL6. And it also can upregulate the expression of the SOCS1. And another example is targeting the signal pathways, the ROS-ERK and alpha-kappa B, and it is a kind of polysaccharides. We called it the APS, the astragalus polysaccharides. It is a kind of macromolecules. It can significantly inhibit the ROS overproduction induced by the palmitate, and it can downregulate the expression of the phosphor ERK, and it can upregulate the AKB alpha and decrease the NF-kappa B translocation. So that's the mechanism on this kind of polysaccharides. Why they can be used to treat the diabetes. Also, another action mechanism is about the modulation of the gut microbiota. And we know that maybe they can be used to modulate the gut metabolism. They can degrade the chlorine into the methylamides, and maybe they can be used to modulate the composition of the gut microbiome. They can increase the content of the beneficial bacterium, and they can be used to decrease the content of the opportunistic pathogen. And you can see here is a scheme diagram showing the main functions of the gut microbes that had a predicted type 2 diabetes association. Here are some examples of the applications of the network pharmacology for the diabetes therapy. First one is about one traditional Chinese medicine that is Gen Qinlian decoction formula, and it is composed of several species traditional Chinese medicine, and then we use the network target analysis, and you can see here we can get the results. There are 13 FDA-approved anti-diabetic drugs from the GQD were predicted, and which supported by at least one independent item of the evidence, and the herbal ingredients in GQD with potential synergistic anti-diabetic effects on berberine in Huanglian were determined. And then the key biological process involved in type 2 diabetes were regulated by different ingredients from the GQD. And another example for the application of the network pharmacology for the diabetic therapy is the Tang Mingling pill. It is developed by Tesla Pharmaceutical Corporation, Tianjin, China, and it contains 11 Chinese herb medicines. And also, we can use the molecular docking and network targeting analysis. And we found that more than 100 active compounds of the 667 compounds in the formula would target 37 target proteins related to type 2 diabetes. And the drug target association network and the drug association network were constructed. The following are some research progress carrying out in our group. First one is about the hyperglycemic activity of T polysaccharides and the T polyphenols. As we know that these are the two main components of tea, and we found that they have the hyperglycemic activity. We use the in vitro and the in vivo BIOSES to determine the activities of these two components. First, to use the alpha amylase inhibitory and the alpha glucosidase inhibitory BIOSES to screen the bioactivities of the T polysaccharides. We found that the T polysaccharides from the black tea had no significant inhibitory on the alpha amylase with low TPS concentration, but showed slight inhibitory with high concentration. And the TPS from the black tea had a significant inhibitory on the alpha glucosidase with increasing TPS concentrations. And here are the molecular docking studies on the bioactivities of the T polyphenols. We found that there is the binding ability of the T polyphenols on the alpha glucosidases, and they have the binding energy and the numbers of hydrogen bonds of 7, and they can bond each other based on the amino acids residues of the following amino acids, such as the SPs 325 and the 304, etc. And we also do the molecular docking on the T-main components, the EGCG, that is one polyphenol of the T, and we study the bonding activities of the critical cytokines, such as the AKT in the signal pathways, and we also found the bonding properties. And we also studied the general physical chemical properties of the T components. The T polysaccharides, we studied the isolation, the structure identification, and the elucidation. And then we studied the activity and the structure and the components relationship. And we have published these results, and it has been cited more than 240 times. And also, we have studied the activity and the cytotoxicity of this kind of components, the T polysaccharides, and we found that it has the antioxidant and the hyperglycemic activity, and with low toxicity. Also, we studied the pretreatment of the coarse green tea, and then we developed the new method to have the high efficiency extraction of these bioactive components, and then to study the structure information and the network. Because it is a kind of macromolecules, we use the AFM and the slightly lysine to study the structure, the conformation information of the T polysaccharides. And because we know that tea can be divided into different types, such as the green tea, oolong tea, and the black tea, based on the fermentation. So we also compared the components of different categories of the tea, and we compare the bioactivities and the structure information of this kind of tea, and to find some rules. And also, we studied the hyperglycemic effects of the T polysaccharides in vivo, and we use the diabetic mice and then to study the related mechanism. So you can see there is the involvement of the PI3K AKT signal pathway in the hyperglycemic of the T polysaccharides on the diabetic mice. Besides the T natural sources, we also studied different type of the natural sources for the hyperglycemic activity studies. This example is the research results on the Inonotus obliquus. It is a kind of fungus. We can see that it can also be found have the anti-diabetes and anti-inflammatory activities. So here are the experimental design, and we also studied the general biochemical analysis and the organ tissue pathological histology studies, and also the related mechanisms. And then we studied another natural source, that is the corn silk. We also studied focused our study on the macromolecules such as the polysaccharides, and also the small molecules such as the flavonoids. We studied the related structure characterization and the hyperglycemic activity of them. Then we found that the corn silk polysaccharides could be a good candidate of the functional food or medicine for the type 2 diabetes treatment. And we found that it can increase the uptake of the NPDG, and it also had the potential to promote the uptake of the glucose in the L6 cell myotubes. And then is about the related mechanism. We can also see that it can modulate the PI3K AKT pathway. Here is the results on the corn silk polysaccharides. They can enhance the GLUT4 translocation instead of the gene expression of the GLUT4. So here are the proposed mechanism for the glucose uptake via the activating the PI3K AKT signal pathway of the corn silk polysaccharides. And another example is on the Inonotus obliquus. It can also have the hyperglycemic activity based on the modification of the structure on the polysaccharides. We combined the chromium with the polysaccharides and to detect the related mechanism. And we found that if we modify the polysaccharides with the chromium, and it will increase the bioactivity. And another example is the black soybean seed coat. We know that it is rich of anthocyanidins, and we also studied the structure and the anti-diabetic activity on the STZ-induced diabetic mice, and we also found it have the related activities based on different biochemical studies and the tissue studies. So let's come to the conclusions and the perspectives. Diabetics, it is a kind of complicated metabolic disease, and it is a current health problem significantly mobility and mortality worldwide. So as a novel perspective, the network pharmacology, it is becoming more and more important tools in the drug discovery in the diabetics due to its multi-compounds, multi-targets effects. And the natural products and the traditional medicines have vast structural diversity and various bioactives, which can provide the opportunities to find different lead compounds and different targets for the diabetes. And although the network pharmacology of natural products is still in its infancy, and the appropriate use of the network pharmacology approaches may initiate new directions, overcome the disadvantages of current anti-diabetic therapies, as well as can contribute new insights into the discovery of the novel anti-diabetic drugs. Acknowledges I'd like to thank the grants from different from the NSFC, the most and the different grants for the supporting of the research of my group. And also, I'd like to thank my School of Pharmaceutical Science and Technology, Tianjin University, and also our Tianjin University Health Science Platform, and all the group members of my lab. Here are some group members of my lab. Okay, thank you. That's all.
Thank you, Prof. Dr. Chen, for your keynote address. Again, thank you to both of our keynote speakers for today's session, Professor Dr. Doralyn and also to Professor Dr. Haji. We look forward for further discussion and collaboration in the near future, where both of you address almost similar topic, but only the difference is the source. Slightly different in terms of as being mentioned by Prof. Doralyn just now. She and her group focus on the marine products and also from marine resources, while from Prof. Hazia's team, you focus on the terrestrial resources. All right. So with that, with Prof. Chen keynote speech just now, so it is the end of our keynote session for day one. Before we forgot that the organizer invite both of our keynote speakers to join the parallel session that will be conducted in a few minutes. So we hope that the PRC for both speakers can share the link with them. With that, tomorrow, don't forget tomorrow we will have another 2 keynote speakers from Aligarh University, India, Professor Dr. Asad, and also from UMT, Professor Dr. Aznawati. So, ladies and gentlemen, we hope that you will have a pleasant day and that you will gain much from this seminar talks and presentation. And today and tomorrow will be a wonderful day for all of you, and do enjoy yourself. Our seminar session will take place concurrently in 3 different rooms based on health, food and environment. To all participants, you may leave this main hall and join each WebEx link based on team, which sub-team session 1 will be hosted by Dr. Razifah Muhammad Razali, moderator Dr. Siti Nur Khadijah Adis, while sub-team number 2 will be hosted by Dr. Fazila Arifin and moderator Dr. Suzana Misbah, and the last session for sub-team number 3 will be hosted by Dr. Siti Aisyah Razali and moderator is Dr. Azila Adnan. So thank you again and have a good day. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.